Okay, today in Honors Biology, I'm going to go over the notes in Kingdom Protista, Chapter 19. I'm going to go through the notes on protozoans and algae. So this is your Part 1 Protozoan Study Guide, or Protista Study Guide, and your Part 2, the algae. And keep in mind that currently this kingdom is really no longer recognized, but your book still has it as a kingdom, so we'll approach it that way. And when we see each other again, I'll give you a handout on supergroups and which of these members of this kingdom belong to what supergroup. Okay, so this is the most diverse kingdom of organisms, so it's kind of like it's got a little bit of everything in it. It's got photosynthetic organisms and those that can't photosynthesize and those that can move and those that can't move, and some of them have cell walls, some of them don't, so it's really very diverse. They are very different from each other, so many are more closely related to members of other kingdoms than to other protists, and that's probably a reason that they've dismantled this kingdom. It includes your protozoans, your algae, your water, and slime molds. So we're going to talk a little bit about each of these in this lesson. One thing that they all have in common, so everybody past our two bacteria kingdoms will be eukaryotic, and so that is a term you should be very familiar with. That means they have a nucleus and membrane-bound organelles. They will contain organelles such as mitochondria, vacuoles, and Golgi. They make up something called plankton, which is the basis of the aquatic food web. And there are over 200,000 species. I never expect that you know the number of species of any kingdom. I just think it's interesting to know that it's a very large kingdom. They can produce asexually by binary fission, which they simply split into two, or by multiple fission, which they can split into more than two cells. Some of them can reproduce asexually by budding, where a new cell grows off of the original, and then some by sporulation, where they form specialized cells called spores that can develop into new individuals. So their asexual reproduction is very varied. Okay, so let's talk about your protozoans. So your protozoans are animal-like. So hopefully we'll have a lab where you actually see some living protozoans so you can observe them moving. This is common to all your protozoans. They are heterotrophic, meaning they cannot make their own food, and they have no cell wall. They're microscopic, so mainly unicellular, although a few may form colonies. Their ecology, most are free living, so that means most of them are not going to harm you if you were to swallow them in lake water, but they're going to require a moist to wet environment. The protozoans make up zooplankton, so one of your forms of plankton, which are single-celled, free-floating, heterotrophic organisms. A few are parasitic, and they'll live in tissues and the blood of their host. They can form a cyst, which is a hard protective covering during harsh conditions. Okay, let's talk about the protozoan structure. Once again, they have no cell wall, so that's why they're animal-like. Their plasma membrane allows for diffusion of gases. So remember, since they're single-celled, they don't have any other way of getting things in and out. So plasma membrane allows for diffusion of gases, nutrients, and waste. They will contain something called a contractile vacuole. So it's a vacuole that holds water, and then once it gets so full, it moves over to the plasma membrane and pushes the water out, so it regulates their water concentration. They can respond to stimuli such as light using eye spots, which contain localized pigments. So if we see the living ones and you see Euglena,'s eye spot is really visible. Okay, there are going to be several phyla, and they're put in their phyla based on their means of locomotion. So remember, we're in Kingdom Protista, so then the next thing was phylum. So this is your phyla. And some of these are going to have more than one phyla name. So your first phyla is called Sarcodina, which is kind of the old phyla name, and I think now it's more Rhizopoda. I usually go with Sarcodina. This includes amoeba, which is a bottom-dwelling scavenger. So that simply means when you go to make a slide of it, you've got to be sure and draw from the bottom of the jar. They're very flexible. They can change their shape. They move by pseudopods. Pod means feet. Pseudo means false, which are temporary cytoplasmic extensions. And their cytoplasm has two regions. They have the ectoplasm and the endoplasm. So the ectoplasm is just inside of the plasma membrane, and then the endoplasm makes up the interior of the cell. So there's a picture of amoeba on page 560 of your text. And the endoplasm can flow outward and form the pseudopods, and then at the same time, another pseudopod can be retracted, and so that's amoeboid movement. So it constantly changes its shape, although slowly. They use phagocytosis to bring in food, so their pseudopod will wrap around food. The cell membrane or plasma membrane forms a food vacuole. Then enzymes from the cytoplasm will enter the vacuole and digest the food. They can reproduce asexually by binary fission. Parasitic ones include entamoeba, which can cause amoebic dysentery. So if you ever drink water from a pond or something and you get sick, it could be entamoeba. And the three of them form multi-chambered shells called test. They are the foraminiferae, which have the shell of calcium carbonate, radiolarians, which are marine, live in salt water. Their shell is of silica. And then the heliozoans are the protozoans that can live in fresh water and form a test or shell. They will have pseudopods, and their pseudopods will extend through the holes in the test. These organisms are sometimes put in their own phyla called foraminifera. So some of this, things like foraminifera, would be a bonus on the next test. Okay, your next phyla of protozoans are Ciliophora, and this includes a genus called Paramecium. Paramecium caudatum is who you will see in lab. They move by many tiny little hairs on the outside of their cell membrane called cilia. So these are short hair-like structures that line the cell membrane and beat like oars, so they all beat in the same direction. They are covered with a pellicule, which is a protein covering that helps maintain their shape. And they have two types of nuclei. They have a macronucleus and a micronucleus. The macronucleus will control cell activities like respiration, digestion, protein synthesis, and asexual reproduction. And then the micronucleus, they may have many of them, and they're involved in sexual reproduction. Since they have multiple micronucleus used during conjugation, this also allows them to have genetic variation. And they will have a funnel-like oral groove lined with cilia. Their cilia will beat. It causes water currents to sweep food into their oral groove, and then that forms a gullet, which would form a food vacuole. So your book would have a picture of a paramecium, and it should have these things labeled. Now, if you see over to the side, I've got SAR, and these are the billates. This is like one of your supergroups, so it's the SAR supergroup. And they're going to reproduce asexually by mitosis and sexually by conjugation. Then your third phyla of protozoans are Zomastogena, which are also called Mastogophora, which are also called Clinoplastidia. They have a single large mitochondria that contains an organized mass of DNA called the chinoplast. That's where they get the Clinoplastidia. They will contain 1 to 2 long whip-like tails called flagella that are used for movement. So your paramecium use cilia, these guys use flagella. The flagella are extensions of the cytoplasm and composed of microtubules surrounded by the plasma membrane. Parasitic ones include trypanosoma, which lives in the blood of its host, and it's transmitted by the tsetse fly, and it causes something called sleeping sickness, which includes fever, swollen lymph nodes, and it can invade the brain. And they're going to belong to the supergroup Excavata. And then Giardia causes giardiasis, which is carried by beavers and contaminated water. And symptoms of giardiasis is fatigue, diarrhea, and cramps. So if you ever drink water from a stream and it's not been treated, you could end up with this little issue. And then there's an interesting member of this phyla called Trichonymphia, which is a chinoplast that lives symbiotically in the guts of termites, where they provide enzymes that help the termites digest food. And then our last phyla of protozoans is Sporozoa, which is also called Amphicoplexia. And these are interesting in that they have no means of locomotion, and they're all parasites. So they're all parasitic forms that produce sporozoites, which are infectious cells that have tough outer coats, which are transmitted during reproductive cycles. And they're also a member of the SAR supergroup. Toxoplasma is fatal to newborn humans. Its host can be a cat or a human, and so that's why they often tell a pregnant woman not to dump the litter box of a cat, because you don't know for sure not if your cat would have this particular protozoan. Probably doesn't, but just to be safe. And then Plasmodium causes malaria, which is transmitted only by the female Anopheles mosquito. And this protozoan will reproduce in the liver and the red blood cell of humans. Each outbreak can destroy up to 40% of that person's red blood cells. And they can release of the toxins, and the red blood cell destruction produces fever, anemia, liver, and spleen damage. So some third world countries are still dealing with malaria quite a lot. Okay, so that is it. That will take you through your Part 1 study guide. Okay, next up, this second part of your protistians are your algae, which are plant-like. So they're going to be autotrophic, meaning they will contain chlorophyll of one type or another and other photosynthetic pigments. They're going to be put in their division. Since they're plant-like, we use divisions, not phyla, based on their type of photosynthetic pigment, how they store food, and their cell wall structure. Some are unicellular, some are multicellular. Most are aquatic, and if they live on land, they must have water for fertilization. Okay, their ecology, they're important because they produce about 1/3 of our oxygen. They form part of the basis of the food web, so they're another type of plankton. So instead of zooplankton, these are called phytoplankton. So these are unicellular autotrophic algae and cyanobacteria. They will live near the surface of water, so they're going to need sunlight for photosynthesis, and they serve as food for aquatic organisms, and they also produce about 1/2 of the oxygen in our atmosphere. Dead algae sink to the ocean floor and over long periods of time can produce crude oil deposits. Diatoms are a type of algae that we'll talk about in a second in their division. They're unicellular algae with a 2-part shell containing silica, which is a glass-like substance. You'll be seeing diatoms in lab on slide. And the dead diatom shells on the ocean floor are harvested to make detergents, fertilizers, and insulators. Then you have another type of algae called dinoflagellates that produce something called red tide. And when they produce a red tide, they release a toxin that can cause paralysis in vertebrates. Like if you eat shellfish that has this toxin in it, then it can make you very sick. And then they're used in cosmetics, ice cream, and cheese. Structure of your algae, they do have a cell wall. They will contain chlorophyll A along with other pigments. Often they will contain something called pyrenoids, which are organelles that synthesize and store starch. Their body is called the thallus. So when we go to lab, you'll be identifying the different types of thallus that these algae have. So here are your forms. Unicellular, these are algae that are just single-celled, and they typically have a flagella. Colonial, these are single independent cells that move and function together as a group. They typically make a little ball. Filamentous, their cells are in a linear arrangement, so they're just a bunch of long cells put together. And then thalloid, their cells branch, and they will have modified root, stems, and leaf, which are called the holdfast. So the holdfast is like their root, the stipes is like their stem, and the blade is like their leaf. And then Euglena is a unique algae in that it's both animal-like and plant-like. It has chlorophyll and it can photosynthesize, so that's why it's plant-like, and it can move and has no cell wall, so that's why it's animal-like. So it can be both autotrophic and heterotrophic, so we sometimes say it's mixotrophic. Reproduction of algae, if unicellular, they can reproduce asexually by mitosis since they have a nucleus, but they can also reproduce sexually. Chlamydomonas and diatoms will undergo asexual and sexual reproduction. If they're multicellular, asexual, once again, is by mitosis, just like unicellular. Sexual can be by conjugation. For instance, Spirogyra will conjugate. And then Odygonium will produce an egg and a sperm. Okay, so algae divisions. So a lot of this algae division stuff that we're going to go over would be like bonus on a test. So the division Chlorophyta, so chlorophyll is green, so these are your green algae. Their RNA sequence indicates they are most closely related to plants than other algae. They will contain chlorophyll A and B, and only plants and green algae have chlorophyll B. They're mainly freshwater, and they form what you see in the spring, what you would call pond scum. And they're going to store their food as starch. They are thought to be ancestors to plants because both of them store food as starch. They both have cell walls containing cellulose, and they both have chlorophyll A and B. Single-celled ones include Chlamydomonas and Desmond's. Colonial include Gonium and Volvox. You'll see Volvox in lab. And filamentous includes Spirogyra and Odogonium, which you'll see these two genus in lab. And they'll have, and then the thalloid form is called ulva. Then you have division Phaeophyta, which are your brown algae, mainly marine. They're going to store food as oil. They'll have chlorophyll C and the brown pigment fucoxanthin. All are multicellular, and they can become quite large. They will have blades, stipes, and holdfast, and they make up kelp and seaweed. Division Rhodophyta are your red algae, mainly marine. Store starch. I misspelled that. I'm sorry about that. Multicellular, and they also make up seaweed along with Phaeophyta. They grow deeper in the ocean. They can grow anywhere from 150 to 270 meters below, because in addition to chlorophyll A, they have phytocythurians, which absorb violet, blue, and green light, which can penetrate deeper. And then you have division Basilorophyta, which sometimes is called division Chrysophyta, which are your golden brown. These are marine and freshwater. They store their food as oil. They include diatoms, which you will see in lab, and these are unicellular or colonial and no flagella, and they have silica in their walls. They will contain the pigment chlorophyll C. They are an essential part of phytoplankton, and they're big producers of oxygen. They have a two-part shell called a valve. We mentioned that earlier. When the diatom dies, the shell does not decompose but sinks to the bottom of the ocean or lake and becomes part of diatomaceous earth, and that's collected to make fertilizers, detergents, and insulators. And then division Dinoflagellata or Pyrophyta. These are the fire algae. They're mainly marine. They store food as starch. They are unicellular but have two flagella, and they're an important part of plankton. They're sometimes called fire algae or dinoflagellates because they can cause the water to sparkle at night, but they also cause red tide or an algal bloom because of an excess growth of the dinoflagellates. And when they are in that number, they can release toxins that can cause respiratory paralysis in vertebrates. Some of them can produce light, so we say they're bioluminescence. Division Euglenophyta, mainly freshwater, so we mentioned these guys in Protozoans. They store food as starch called perimylin. They're unicellular with flagella. Their body's covered with the pellicule. They're similar to both green algae and protozoan because they contain chlorophyll A and B, so can photosynthesize. So that's why they're like algae, but they have no cell wall and move due to flagella. That's why they're like protozoans. So some of this is kind of repetitious. Okay, then you have the fungal-like protist, which can move during some part of their life cycle, and these are all decomposers, so they're important to breaking down material. You have two main groups. You have slime molds and water molds. So your slime molds include your Myxomycota or the plasmodial slime mold. So stuff like this is like what would be a bonus on your test. They're composed of a very large cell with many nuclei and a lot of cytoplasm, and they can move across the ground absorbing decaying matter. And then you have the Ascomycota or cellular slime molds, which usually hang out as individual cells but come together in one mass if their food supply is short. And then finally you have your water molds, which include the phylum Oomycota or Oomycetes, which are filamentous and stationary, so they do not move. They can be decomposers, they can be parasites of fish, and they're responsible for the potato blight in Ireland in the 1800s. So hopefully this can help you get through your notes to fill in that you already have in your possession, and then you're going to work on your two study guides, Protozoan Part 1 and Algae. Thank you.